Hello, this is the prep for lecture 19, which we'll probably be starting tomorrow. And so I apologize for not having it ready sooner. I, for some reason, had in my head that I'd already done these. Yeah, and so I was working on lectures 20 and 21, and I suddenly realized, yeah, no video. So hopefully this won't be too late for you. And what we're going to look at in this lecture, which goes on for a fair length of time, so we will definitely be taking up a lot of Friday's lecture too, but we're looking at modified dihybrid ratios, and we're going to look at lots of different dihybrid crosses that give those 9:3 to 3:1 type ratios, but then the phenotypes are not necessarily what you would expect. There'll be sometimes 9:3 to 3:1 ratios that are just all different variations of the same phenotype, but sometimes there will be interactions, things like epistasis, when being homozygous or heterozygous for a certain genotype means we can't actually see what the genotype is doing at the other locus, and things like that. So we're going to look at lots of different ways multiple genes interact. So basically, when people talk about non-Mendelian genes, they're really not talking about anything about the genes that's, oops, that didn't show up right away. They're not really talking about anything weird about the genes. They're simply talking about the way the gene products are contributing to the phenotypes. And so this is you should keep this in your head, because this is the ratio we're going to be working with this whole lecture. And so, if this was, in plants, a certain flower color and then a certain pollen shape or something, you'd expect to be looking at, either the purple flowers or the white flowers and the long pollen versus the round pollen or something like that. But in some cases, there are just 4 different variations of the same phenotype, or sometimes you will get 2 of these categories that combine and they give you the same phenotype. In a couple of cases, we don't even know quite how it works, but if there are places where we do know how it works, I will try to be clear about that. Okay, so here's a situation. So we have some chickens here, a female chicken that has what is called a rose comb. I have no idea why it's called this. And another parent, the male, who has a pea comb. I can tell why that's a pea comb. It looks like a pod that's open with the little peas visible. And when these two are crossed to each other, all the offspring that they produce have what is called a walnut comb. It sort of has a walnutty appearance. So here's a situation where our heterozygote, our F1, and remember, when we're doing these crosses, we always assume the parents are pure breeding unless you're told that they're not. So in this case, we have 2 things that we could be considering. In one case, we could be looking at, some form of codominance. This doesn't look at all intermediate between the 2, but if you take sort of, the shape here and the shape, maybe in some way these things are working together to produce this funny phenotype in the heterozygotes. So that would be a single gene, right? So this individual would be like this, and this would be like this, and then it would turn out that the heterozygotes have this unusual phenotype because the 2 alleles are sort of independently altering that phenotype. But it could be 2 genes, and this could be complementation, right? You could be mutant for one gene and wild type for the other, and the other parent could be wild type for this one and mutant for this one. And so then when we look at the heterozygote, we're going to have complementation, right? You have one good functional copy of gene B and one good functional copy of gene A. So these are the 2 things that we're thinking about, and we're going to consider the possibilities for both in the class. So consider what information we need to determine if this is actually our correct outcome. And you might stop and then say, what would you do? Would you cross these to each other, for example? That might be something to consider. Okay, so what do we need to know? If we're trying to determine if it's one gene or two, we need to start with what is wild type, because if there's a wild type phenotype and it happens to be that walnut comb, wild type is not going to be heterozygous because it's the commonest one found in nature, and you can't maintain that, right? If you're heterozygous, you're going to give rise to the other genotypes whenever you mate with another heterozygote. So wild type is not due to heterozygosity in general. Can we make a pure breeding strain of walnut comb chickens, right? Because as we saw with the lethal alleles with the yellow mice, for example, you can't make a pure breeding strain because you have to be heterozygous in order to have that yellow coat color. So if the walnut is due to being heterozygous, you know, big R, little r, for example, then you can't make a pure breeding strain of it. So that's another question. And then finally, what happens if we cross them to each other? If they were heterozygous, what would you expect to see? You'd expect to see some with the, whatever that first one was, the rose comb, and some with the walnut, and some with the pea comb. And you should say how many you would expect to see for each of these. And then, but if we're talking about two genes, if this was wild type, that would make sense for two genes. And if it's two genes, we could make a pure breeding strain of it. And if we cross the walnut comb F1 among themselves, we won't get this, you know, 1 to 2 to 1 kind of ratio. We'll get a different ratio. So think about the consequences. If you're trying to determine whether there's a single gene or two genes, this is the kind of information we need to know, and we need to answer all these questions in order to help us decide. And it turns out in the F2, we basically end up seeing 4 different phenotypes, and they are 9/16 of the walnut, and 3/16 of the rose, 3/16 of the pea, but there's this new category called single comb. And if you look at these ratios, you can figure out how the genes work. So a walnut individual with the walnut comb has the dominant alleles for both genes. The rose comb, they are mutant for one of the genes but wild type for the other. The pea comb, they're mutant for the other gene but wild type for the second one. And the single one is mutant for both of those genes. Okay, so when you look at this ratio, you know that you can't have a walnut comb based on heterozygosity for a single gene. If you have 2 alleles of a gene and you cross these to each other, there's no way you're going to get 4 different phenotypes out of that, right? There's only 3 possible genotypes, so there can't be 4 phenotypes. So have a look at that and just make sure that you understand how that worked. And the appearance of this new phenotype is one of the, first of all, the ratios, of course the ratios, but also another piece of evidence that we're talking about multiple genes is this appearance of a 4th phenotype. Okay, so this is just a message thing which you can read on your own. And so there's another example here, which is corn snakes. Corn snakes are quite beautiful, actually. They have synthesis of both orange and black pigments that are laid down in a particular pattern that helps them stay camouflaged. They tend to be slithering around in the leaf litter on the forest floors and so on. And so they're well hidden when they have the camouflage. But you can also make pure breeding orange strains and pure breeding black strains, each of which have only part of the pattern. But when you cross the 2 pure breeding strains, you get the wild type. Okay, so therefore we know how many genes are involved, and you should be able to, you know, given just this information, you should be able to deduce. This is enough information for you to figure out what's going on. So this suggests to us then that we have 2 different genes. The black ones are mutant in the gene that is responsible for making the orange pigment, but they're making lots of the black pigment. The orange ones are making orange pigment with their wild type orange gene, but the black gene is mutated, so you don't get any black pigment. And then when you make the F1, they're heterozygous. And so they have that camouflaged phenotype. And so you should ask what would happen if you cross these to each other and what kind of ratios you would expect. And we're going to go through some of it in the class. So this is it's quite pretty actually. This is the camouflaged one. Here's the orange one that's mutant for the black gene, but wild type for the orange gene. And you can see the sort of the whitish parts of the pattern, but there's no black pigment. And then this is the black one, and so it's got black pigment and the white, for the stripy pattern. And then the albino still has those funny white stripes, but there's no pigments of any type made. And so because the skin is pretty thin, you're seeing this sort of pink transparent almost snake, an albino. And these would be the genotypes. Okay, so these previous slides, they're just talking about how two different genes combine to affect the genotypic ratios. That should say phenotype. I don't know why I have genotype there. But they're not directly interacting. So they're kind of like two separate pathways that are independently each contributing something to your overall phenotype. However, there are types of interactions where the genes are not independently contributing. They might be working in the same pathway, for example, where one acts before the other. And then we could see examples of epistasis, which are these ones I'll talk about in a bit. Sometimes we'll see examples of what is called duplicate recessive epistasis or complementary gene action. And I'll show you an example of that. Duplicate gene action, a bunch of different kinds of interactions. And I'm not as concerned necessarily that you memorize the names of these things, unless memorizing the names helps you understand how they work. If you can explain how this works, recessive epistasis, that is, the gene responsible for this phenotype acts before the gene that's responsible for this phenotype, but they're acting in the same pathway, then that's good enough for me. I find there's very little value in you just memorizing these terms unless you actually understand how they work, because I'm never going to ask you, just tell me the memorized name for this ratio. I'm going to ask you, how does this ratio come about? How do the genes interact? Show me the 9:3 to 3:1 and tell me how these things work together to produce, blah, 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 the phenotype. So just I'm trying to be clear about sort of the expectations there, because I find that a common problem is students just not understanding what we're asking them to do. And we're not asking you just to memorize this stuff. We're asking you to understand how it works and be able to explain in your own words how that works. So there's many examples of epistasis, and literally, and this is Latin, epi means on and stasis is standing. So it's kind of means standing on. And it's the interaction of two different genes where the genotype at one of the genes may prevent us from being able to tell what the genotype is at another locus. They're often involved in the same pathway biochemically, or sometimes in the same developmental pathway as well. And so this is an example. Suppose you cross two plants and there's pink flowers and white flowers, and then you have blue flowers in your progeny. This doesn't look like sort of any kind of intermediate phenotype or codominant, so this must likely be, complementing genes. There's two different genes. And so if we cross these to each other, we do get sort of a 9:3 to 3:1 ratio, but one category is the combination of two. So hopefully you can see that 4/16 must be 1/16 and 3/16 combined. And so 9/16 would be, two dominant alleles. The 3/16, you'd be mutant for one of them, but have at least one dominant for the other. And then these ones would be mutant for the other gene, the B gene, and mutant for both genes. And when we see this, this is an example of recessive epistasis. And what it means is that the gene that encodes this enzyme, if it's homozygous mutant, this enzyme can't perform a biochemical reaction. And that means even if the plant, in this case, has the enzyme for the other reaction, it doesn't matter because the whole process is cut off at this step. And I think I have a, I think I have another an explanation coming up. Yeah, so you've basically got two different enzymes, W and M in this case, and we use the arrow to indicate where each of those is functioning, and this is the genotype. So as long as you have one dominant allele of the W locus, then you're going to have enzyme 1 present. It's going to convert this substance into a substance that happens to have a kind of a pink color. And if the M enzyme is functional, it converts that into a blue pigment. And so you have these wild-type blue flowers. If the M gene is mutated, there's a block in this pathway. The enzyme is not available to make this conversion, and so what we get is an accumulation of this pink pigment, and the flowers end up being pink. However, if we're mutant for the other enzyme, we basically block the pathway at this first step. This first reaction is the one that can't be catalyzed. And so even if you could have the wild-type enzyme for this step or not, but it doesn't matter because you're not producing the pink pigment, then it doesn't matter if you can convert it into the blue or not, because there's not going to be any pink pigment at all. And so therefore, a quarter of the progeny are going to have this genotype. And when we see that 9:3:4 ratio, we can tell which of the genes is acting first. And in this case, it's the one that encodes the enzyme for this step. If it turned out that pink was the color of this intermediate and white was the color of this intermediate, because it doesn't have to start out colorless, then we'd have 4/16 of the pink and only 3/16, or sorry, 4/16, yes, of the pink and only 3/16 of the white. And so just hopefully that's clear. I'm just going to get rid of that weird slash that I just made. Okay. So that's what recessive epistasis is, and basically it just shows you like where, it lets you see where the pathways are blocked and why you get that ratio, and that's the thing we really want you to key into. Why is this ratio being produced? And I'll quickly go through, let's see, yeah, I think I have time to go through this one before I do the next video. So basically this is how fly eye color works. There are several genes that are required to encode the enzymes that encode the production of a brown colored pigment. And there are several genes. One of these is made up, the BN, brownish, because it's a little more complicated on the orange side, and I just want to keep this part of it simple. Basically, you have several genes that are required to make an orange pigment. If this is mutant, there's no orange pigment and the eye is brown. If this is mutant, there's no orange pigment and the eye is brown. If both of them are mutant, there's no orange pigment and the eye is brown. So this is how you can sometimes get single mutants in a particular gene and then double mutants that give you the same phenotype. Okay, that's what the key will be here. And the same with scarlet and cinnabar. So we make a mutation in scarlet and there's no brown pigment made, so therefore the eyes are going to be orange because the only thing left is the orange. If we make a mutation in cinnabar, orange eyes. If we make a mutation in both genes, orange eyes, and they look exactly the same as each other because they all have the exact same thing wrong with them, which is that they don't have any brown pigment, and so therefore their eyes are actually quite a pretty orange color. And so I'll have you just look at that and think about it, and when I start the next video, then we'll talk about duplicate recessive epistasis.